Hello, welcome everyone to the Arthritis National Research Foundation's Researcher Spotlight webinar series. My name is Ashley Barrow, Director of Individual Giving here at ANRF. Now, on behalf of our Board of Directors, Scientific Advisory Board, and staff, I would like to welcome you all to the Spotlight on Rheumatoid Arthritis. We are excited to have so many of you joining us from all around the world. We would also like to recognize and thank each of our corporate partners for supporting our Researcher Spotlight series. Thank you, Amgen, for being our gold level supporter for this series, and thank you to AbbVie, AstraZeneca, and AOPI for your continued partnership and dedication. It is because of our partnership that we are able to continue to pave the way for new innovations and learnings from our researchers. Thank you. Before we get started, there are a couple of logistical pieces of information we want to share. First of all, this webinar is being recorded and we will have it available on our website and YouTube channel in the coming weeks. If you have questions, please go ahead and type those into the Q&A. If we aren't able to get to yours, please email info@curearthritis.org after the webinar and we can get to those questions. All right, without further ado, I am now honored to turn the mic over to our patient moderator, Nancy Stevenson, who will tell us her story of living with arthritis. Thank you, Nancy.
Yes, thank you. Hi, again, my name is Nancy. My RA journey started at the age of 25. About 2 months after the birth of my first daughter, I woke up one morning to a red, swollen, painful ankle. And this was unusual for me. I was an athlete my whole life. I had been an athlete my whole life, but this pain felt different. But I didn't really give it much thought until the next morning when I woke up and the pain had gone from my right ankle to my left thumb. From there on, it just continued to migrate over the next few weeks. I finally sought out help about a couple months into this going on with my primary care. I underwent many tests such as Lyme disease, parvovirus, rheumatoid arthritis, which of all came back negative. This was very discouraging for me as I didn't know what was going on and I was in excruciating pain. The RA quickly, undiagnosed RA at the time, quickly attacked my hands and feet, and it was relentless no matter what I tried. I finally did receive an RA diagnosis about 2 years into my journey on symptoms, bilateral symptoms from a rheumatologist. I was on anti-inflammatory medications. I then moved to disease-modifying drugs, and now I am on a biologic drug. These drugs have, at different times during my journey, given me significant relief from the disease, even years at time where I really didn't feel like I had RA. And I really treasure those years now because when we have the bad days, we really remember those good days. From there on, I have continued to search and research alternative and complementary therapies to help me deal with my RA, with both the visible signs of RA, such as deformity, redness, and swelling, as well as the invisible signs of RA, which are, pain, excruciating fatigue, anxiety, depression, helplessness, and hopelessness. But I continue to push through, and I do consider myself an RA warrior because 32 years later, I still get up every day with fight in my heart to face whatever my rheumatoid has in store for me for the day. And I continue to seek out help. And I am really excited on that avenue to be here with you today and see what these two researchers have in store and what they're going to share with us and what's new on the horizon in the treatment for those of us dealing with rheumatoid arthritis. And without further ado, I am going to have the pleasure of introducing our first presenter, Dr. Chang. She is a pediatric rheumatologist at Boston's Children's Hospital and assistant professor of pediatrics at Harvard Medical School. She attended the David Geffen School of Medicine at UCLA, where she completed her MD, PhD training studying the effects of galactin-1 on dendritic cell function and immune regulation. She then completed her pediatric residency training at Boston Children's Hospital and graduated from their pediatric rheumatology fellowship program in 2019 before joining the faculty. Her lab studies site-specific autoimmune diseases with a focus on the role of synovial resident memory T cell in joint-specific memory and arthritic flares. And with that, I pass it over to you, Dr. Chang.
Thank you so much, Nancy. And thank you for sharing your story. I think your story, now I'll adjust a little bit during my talk, I think is one that we have heard a lot, and many patients experience, and bravo for continuing to fight. Oh, okay. All right. So a little bit of, oops, hold on. Okay. A little background about me. So I grew up in Southern California in Irvine. Nancy kind of mentioned already that I spent a lot of time in Los Angeles. So I got my Bachelor of Science in Microbiology, Immunology, Molecular Genetics from UCLA, then stayed at UCLA for my MD PhD training, and then moved out to Boston a little over 10 years ago. I wear lots of hats, as we all do. I'm a pediatric rheumatologist. I run a lab, and I'm an assistant professor at Harvard Medical School. I'm a mom to two young kids who are growing, a 7-year-old daughter. Here's the picture of my family, my 4-year-old son, and then a 14-year-old Beagle Terrier mix. So she was our first fur baby. I'm a assistant coach for my doctor's softball team. So trying to do the mom, the scientist, the physician, kind of all at once. Kind of one interesting thing, I guess, about me is I joke that my roots are humble. So my first job after high school, I was the mascot whale for a 4th of July celebration at my first job. And we used to keep on my fridge a picture of me in my whale costume standing next to some random person. And I would say, like, look, that's a picture of me. But yeah. Okay, so a little bit of background about rheumatoid arthritis. So what is it? It is an autoimmune disease that causes chronic inflammation of the joint. And while it mainly affects joints, there are other organ systems that can be involved as well. Heart disease and lung disease are the main ones with RA. Signs and symptoms, morning stiffness, joint pain, joint swelling, the redness that Nancy mentioned. And typically it involves the hands, wrists, feet, and is often symmetrical. And I say that because there's kind of two subtypes of rheumatoid arthritis. One, which we call seropositive RA, where there's antibodies on blood tests, and one that's seronegative RA. And for adults, the majority of patients have the seropositive RA with this presentation. How is it caused? So it's caused by an inappropriate immune system activity that causes inflammation in the joint lining, also known as the synovium, and I'll talk about that in a little bit. And this inflammation leads to structural damage and changes in the joint, as well as fundamental reprogramming of the tissue and immune cells within the joint, kind of making them more inflammatory and more prone to inflammation. So arthritis, we also refer to it as synovitis because it's the synovium, or that lining of the joint, that becomes inflamed. And so this is a kind of cartoon of a normal joint and what an inflamed joint looks like. And you can see that, there's erosive changes, or you start getting degradation of the bone and the cartilage. And so that really creates a lot of pain and mechanical issues. And sometimes patients need joint replacement therapy because of that. Up here on the top right is, so if you took a slice of the synovium and put it on a slide and was looking at it, this is kind of what we're looking at. Here's an edge of cartilage and some bone, cartilage and bone, and synovium. This is normal. And what you normally see for synovium is it's a super thin membrane. It's just a few cells thick, 2 to 3 cells thick. And it lines the joint, and it makes a lot of the joint fluid that keeps things running smoothly, and so that your joint doesn't grind. And when you have inflammation, and now you can see that there's all this purple in here. And so this synovium becomes thickened, right? It's no longer this thin lining, and now it's really thick and huge. And there's also a lot of other immune cells in there, and it just looks like a hot, ugly mess. When synovium becomes thickened, another word that we use for it is pannus. And you can see here in an inflamed joint, it kind of looks like this gooiness that sticks to the cartilage and the bone in the joint, and it actually starts eating away at it. And so this is kind of showing that the synovium is starting to eat away at your bone and your cartilage. And that can all happen from long-term inflammation. And so the mechanisms that promote joint inflammation or how arthritis happens is very similar in adults and in children. In children, we call it JIA, juvenile idiopathic arthritis, and I think there's a webinar on that a little bit later. But so that's kind of how I started down this path. A little bit more information about the disease. So how common is it? It's very common, actually. So the prevalence of rheumatoid arthritis in the United States is about 0.5 to 0.6% of the population. So about 1 in 200 individuals. So if you're thinking back in college and you're in a lecture hall, like somebody in that lecture hall is going to have RA. How is it diagnosed? So history, physical exam, imaging studies are also helpful, such as MRIs or ultrasounds. Blood tests can offer clues. I mentioned earlier, there's a subclass of RA we call seropositive, where you have antibodies in your blood, namely rheumatoid factor and CCP. But blood tests cannot diagnose RA on its own. And so, you know, as we had seen earlier with Nancy's story, that you can have negative blood tests and still have arthritis. And just having a positive blood test also doesn't mean that you have arthritis, because these antibodies can pre-exist in your blood for decades before you actually develop arthritis. And what is the expected course of disease? So RA is a chronic progressive disease that can lead to joint destruction, deformities, and disability. And arthritis flares are very common in RA and can cause the disease to wax and wane. And so sometimes you feel like you're doing really great, and then you'll have a flare. And that's the meat of like what I'm really interested in studying, why these flares happen and what contributes to that. Treatment and management. So how can someone manage the disease? So there's some things that you can, it's a combination of treatment and then having a healthy lifestyle. And so, having a healthy diet, being physically active, avoiding smoking, these are all things that are important. So why? So diet, so cholesterol matters, as there's a 2 times increased risk of heart disease for patients in RA. And so you want to make sure that you have the healthy cholesterol levels. Physical activity is important, because having, if you routinely do cardiovascular exercise or like lose weight if you're overweight, this can help decrease the burden on your joints. And being physically active can also help to maintain your mobility and the range of motion in your joints when chronic inflammation can cause some contractures to form. And then avoiding smoking, why that's important, because there's a link between smoking and increased risk of lung disease and rheumatoid arthritis. And there's also some evidence that some of these antibodies that contribute to RA can be generated in the lungs. And so you really want to have as healthy of a lung as possible. And how do we manage RA? And so what are the treatments? So combination medications and surgery, mostly medications. And so there's lots of different medications that can be used. And if you guys have RA yourself, you've probably experienced going through the different categories of them. So steroids is one that often works quickly, but nobody really wants to be on steroids long-term because there's lots of nasty side effects. Non-steroidal anti-inflammatory drugs, or NSAIDs. So these are things like Motrin and Aleve and Celebrex. DMARDs, which is disease-modifying anti-rheumatic drugs. And so these are things like methotrexate, sulfasalazine, leflunomide, and others. Biologics, and so these are things like Enbrel, Humira, Remicade, Actemra, Rituxan, and many more that I didn't list here. And then a newer class called JAK-inhibitors, or JAK inhibitors, to target the different immune pathway. And examples of these are tofacitinib or Xeljanz or ruxolitinib or Jakafi. And then ultimately, sometimes people need surgery. And so having a synovectomy, where a surgeon goes in and cuts out all that inflamed synovium, can sometimes be helpful in the treatment of arthritis and ultimately joint replacement if there's been a lot of damage that has been done. Early treatment and tight control are critically important to good outcomes and avoiding joint damage and joint replacement down the line. Okay, so what's my focus? And so my focus is on synovial resonant memory T cells, or TRMs, and joint-specific memory. So what is that? Joint-specific memory in arthritis. And so I'm going to start out by saying the statement that there is joint-specific memory in arthritis. And that's basically the concept that once a joint has experienced arthritis, it's kind of never quite the same as again. It's never quite a naive joint again. And so each subtype of arthritis presents with different patterns of joint involvement, but every patient has a unique pattern to them. And the same joints that were inflamed before are predisposed to flaring again, regardless of the type of joint, and that's what joint-specific memory is. And so this is a study that we had done looking at a cohort of pediatric patients with juvenile childhood arthritis, and we followed them through adulthood into their adult and rheumatology clinic, so like decades and decades, and we found that like when they flare, the majority of times that they flare in the same joint that they had arthritis in before. And there's an immunological explanation for this, and that's resident memory T cells. And so what are resident memory T cells? So TRMs are long-lived immune cells that remain in the joint tissue during remission and act as key mediators of arthritis flares and joint-specific memory. And so this is a cartoon of how I kind of generally talk about this. And so you have a normal joint, very healthy, and then something causes inflammation to occur there, whether it's an injury, whether you had an infection or something, you get acute inflammation, so early inflammation. And during that inflammation process, a lot of different immune cells go into that joint to create that inflammation. Some of them are T cells, and then some of those T cells decide to stay. And so when we treat you with lots of medications, with steroids, with whatnot, and we get your arthritis under control, you feel great. But some of those T cells are staying there in remission, despite the fact that you feel great and that you don't have any active joint inflammation. And these are your resident memory T cells. And then if you activate these resident memory T cells, they can cause an arthritis flare. And so hence the cycle of flare, remission, flare, remission. TRMs, and then in some of my earlier work, we had shown that if you, at least in mouse models of arthritis, if we take a mouse that had arthritis and is in remission, and we deplete these TRMs from the joint in remission, then they don't flare again when you challenge them and trigger them later on. And so what does this mean for arthritis treatment? And so arthritis flares are not benign, right? Because each time that joint flares, there's a fundamental change that happens within that joint where it's not quite normal again. And we call that the joint accumulation hypothesis, which is on this graph here. So the blue line is every time you have an arthritis flare, you have some joints are inflamed. And then you go into remission because we give you more drugs, and then you go, and then it comes back down, and then you flare and you have more joints this time. And then we treat you and you go back down. And each time you flare, you notice that you have more and more joints that develop arthritis. And that your joint count for the joints that are in your body that are no longer quite the same again, starts accumulating over time. Right. And each time that you flare, at least in our study, there was a 40% risk of having a new joint become involved that didn't have arthritis before. And you think that if this process, you're generating more TRMs in these joints, and other mechanisms as well, kind of reprogramming synovial fibroblasts, accumulation of inflammatory macrophages, and these are other immune cells that also get fundamentally changed within that joint every time you have inflammation. And so you can think of how that is a bad thing, right? That if you flare, you get more joints involved. And so our goal is to understand how TRMs develop in the joint and to help us design new therapies to mitigate this joint accumulation in arthritis flares. And so this is kind of a cartoon. One is, what are the factors that promote TRM formation in the joint is one of the interests in my lab. The other is what are the factors that keep them there? So why do they stay there so long? And then how do we selectively target these cells so that we can kind of reset that joint for the future? To help study that, and so to kind of the first question, how did TRMs form in the joint? We use several different models. One is using mouse models of arthritis, another, but no, those are mice and we are people. And so now we also developed a human synovial organoid, which is basically like we're recreating the tissue in our lab. And so, tissue has 3D structure, it's not just the 2D image. And so we want to create a 3D structure containing all of the cells that you would normally find in synovium, as well as these T cells, and kind of to model how TRMs form. And so this is just a picture of what that little organoid looks like. It looks like a little ball. And then if we look at the different cells within them, there's the fibroblasts that form a lot of the structure. There's also blood vessels that go into the synovium, and this is them starting to form blood vessel cords. And then the T cells are kind of scattered in between. One of the things that we found, that exposure to inflammation increases the number of TRMs that are generated in our little synovial organoid model. And TNF, which is tumor necrosis factor is one of these cytokines that are produced during inflammation. And one of the things that a lot of these are biologic drugs target. So these can actually stimulate fibroblasts to produce factors that mediate TRM development. One of the known ones is IL-15. And so my supported research from ANRF is to test various strategies to interrupt TRM formation in the joint and with it future arthritis flares through targeting IL-15 production in our human organoid and mouse arthritis models. Okay, so quick summary. So rheumatoid arthritis is an autoimmune disease that causes chronic inflammation of the joints that leads to damage and disability. Inflammation causes joint-specific memory, a propensity for the same joint to flare again, and this can accumulate as the disease burden grows. Arthritis flares and joint-specific memory are mediated by resident memory T cells in the synovial tissue, and targeting TRMs can attenuate local arthritis flares, at least in our animal models. We have developed animal arthritis models and human synovial organoid models to study TRM development in the joint, and that early studies of ours identified factors produced by the synovial tissue, so by the joint tissue during inflammation, that can promote TRM production and formation, and we're currently testing strategies to interrupt this pathway. So special thanks. So this is my small but mighty lab, the Chang lab, and our collaborators. So Peter Nigrovich is a mentor of mine, and he's where I started working on this project of arthritis flares and TRMs. Brian Woffer and Chang are people who used to be in the Nigrovich lab, but now has since moved on, and a lot of different collaborators have really contributed to this or work in our ability to study arthritis. And then these are our supporting funders.
Thank you, Dr. Chang. You brought a lot of clarity to my trajectory of my disease over the past 32 years.
Yeah.
And as to why my feet and hands were predominantly taken out first, if you will, but then moved to other joints. And I never understood that. So you brought a lot of clarity to my disease trajectory over the past few years. But what advice would you give to someone who is newly diagnosed given this information?
Yeah, I would say, and this is something that we're implementing now, at least in pediatrics in our pediatric patients, and is that, you know, aggressive therapy up front. So we want to shut down the inflammation as quickly as possible, and then we want to keep tight control of that, right? Because the longer that you let your arthritis and inflammation kind of brew and linger, the more damage is done, and the more likely that it can spread. And so the other piece of this is, you know, as you mentioned, Nancy, so even when you feel like you don't have arthritis anymore because you're on medications for like 10 years and you feel great, there's always a risk to stop, right? And we see that a lot in our patients, and like, who wants to be on meds and who wants to be a teenager who's on meds and different from all their, you know, their classmates and their friends. And so there's a risk there. And it's something that I have made a priority talking to my patients about the importance of staying on top of our disease.
Yeah, I agree with that. I've noticed that as well. And it's always very complexing as to why it moves to other joints and as to why it can go away and come back. So that's very helpful and hitting it quickly. I appreciate you saying that. And I have a question from an audience member. Do you think these T cells are also present in other types of arthritis, such as psoriatic arthritis?
So absolutely, there's actually a whole body of literature looking at talking about resident memory T cells and their potential roles in psoriatic arthritis, and not just arthritis, other immune diseases as well. And so they're actually a big player in psoriasis, so the skin disease that's associated with psoriatic arthritis. TRMs have also been talked about in terms of being key players in inflammatory bowel disease and other autoimmune diseases as well. And so, you know, they're there for a normal function, as all immune cells are, to protect you from infection. But when they start doing things you don't want them to do, they can cause problems.
Thank you. And just a reminder, anybody listening in, to please type in your questions. We'd love to forward your questions to Dr. Chang. What inspired you to research in this area? What got you inspired?
So, my background, my PhD training was in immunology, and I just find it really fascinating about how the immune system can affect every part of your body. And I'm very interested clinically in complex diseases. And when I started spending time in the rheumatology clinic, it was really this phenomenon where we saw where, a kid comes in with childhood arthritis and they have their left knee and their right ankle inflamed, and you treat them with meds and they get better. And then so you have hope that maybe you can stop the medications and they flare in the same places, which is really interesting because we think of autoimmune disease as being something that affects your whole body. So why these places? So that's kind of how I started down this pathway and started down looking into resident memory T cells and joints.
I have another question from the audience. And what are identified RA triggers?
That's a difficult question to say. I think the things that we talk about in patients, and I don't know how much evidence is behind it, but we usually think about trauma as being a big one. And so we certainly, if someone has an injury, broke a wrist, that's a place that we've often seen arthritis pop up. The other one we talk about is infections. And so sometimes when you get the flu, you feel like your joints are achy. And so we think that, if you get the flu, could it trigger an RA flare? And then the biggest one that's tried and true though is like stopping medications, right? And so that's the majority of the times when you flare is when you stop taking your meds, whether directed by your doctor or not.
I like that you pointed out infections as my arthritis, as I mentioned, started after the birth of my first child. My sister's RA developed after a bad case of the flu. So interesting point. Another question, do you find that taking a disease-modifying drug keeps a biologic working longer?
So there's certainly evidence out there that taking the two together has synergistic effects. And so it's better than each working alone. The other reason why I would advise for people to be on a DMARD is with certain biologic medications like adalimumab, or the common name is Humira, or infliximab, or the common name is Remicade. And so some of these medications, a person's body can develop an immunity to it. And so they actually, it stops working for them. And we find that patients who are also on a DMARD at the same time, that happens less frequently. So it helps the longevity of your drugs working in your body.
Thank you. Are there any new diagnostic methods or tools that could help improve early detection?
So it's a tough question to answer because there's the logistics of, are you going to image everybody, right? And so essentially, we take a history, we do a physical exam. It's only so sensitive. Sometimes when in clinic, when I'm not sure whether or not someone has joint inflammation, then I pursue imaging. And imaging is either ultrasound or MRI. MRI is more sensitive than ultrasound. And so, you don't always want to MRI everybody, and you don't want to MRI them all the time. But those are certainly more sensitive. That being said, there were studies that were shown that even if it seems like your joint is not inflamed on MRI, there was actually a study where then they went and biopsied those joints and showed that actually, if you look at on the cellular level, there is actually still ongoing inflammation. And so there's a level of sensitivity there.
Perfect. Do you have any other questions that you would suggest that we ask our rheumatologists, whether newly diagnosed or we've had RA for a lot of years, but any specific questions that I should ask or we should ask our rheumatologists?
I don't know about if it's a specific question, but I want to say that, RA and arthritis and any autoimmune disease, it's a personal journey, right? And there's always cost benefits to everything. And so one of the things that I do with my patients is we discuss what are important to them. What are the things that are important in their lives that make their lives worth living? And we want to make sure that we keep encouraging those things. And so whatever medications, and maybe this is more true in children than adults, but whatever medications we choose, they have to be on board and be able to take. And so it's a joint discussion, and it's a joint decision. And so I would suggest, that you advocate for yourself. and advocate what's important for you and when something is just not working, because we're, as physicians, we're supposed to be there listening.
That's great advice. Thank you for that. And thank you for your presentation and answering all of my questions or our questions, Dr. Chang.
You're welcome. Thanks for having me.
You're welcome. We'll now move on to Anna Helena Jonsson. Welcome. Anna Helena is a rheumatologist and a scientist. She spent the past 9 years in Boston at Brigham and Women's Hospital and Harvard Medical School studying the role of T cells in rheumatoid arthritis. She recently moved to the University of Colorado to set up her independent research lab as an assistant professor. She will continue to work on CD8 T cells and a special molecule called granzyme K that is found in inflamed joints of people with RA and many other rheumatologic diseases. Welcome, Anna Helena.
All right, thank you so much for that introduction, and thank you to the ANRF for inviting me to speak today. I'm so excited to share this research with you, and also very happy to take questions. So please type in your thoughts in the Q&A, and I'll try to leave a lot of time at the end. So I think everyone who's here knows what rheumatoid arthritis is, and hopefully when you look at your own hands, they don't look quite like this. You know, as a rheumatologist, I'm happy not to see these kinds of hands that often anymore. But this is the kind of thing that can happen when rheumatoid arthritis goes uncontrolled. And even today in 2023, you know, there are some patients who have hands like this or who are on their way to hands like this, because even though we have a lot of treatments available, some of them don't work for everybody. And so that's why, you know, scientists like Dr. Chang and me and so many others, we're trying to figure out more and more about rheumatoid arthritis to identify new treatments. So, so you saw some normal synovium before and some RA synovium. I have a different picture because I think that, you know, as especially as cellular immunologists like Dr. Chang and I are, you know, we're really focused on the cells that are in synovium and figuring out what they are. So here on the left, you see normal synovium. So this is the joint lining that sort of forms a little pouch around each joint that keeps in the lubricating joint fluid that allows your joints to move smoothly. And it's supposed to have this sort of loose connective tissue. It's just kind of like nice, fluffy, almost kind of like bubble wrap. And then you have this lining up here that makes this the sort of the grease that makes the joints work well. But in rheumatoid arthritis, seen here on the right, you have the immune system attacking this tissue. And so there's been a major invasion of all these different cell types. And now it's thick with stuff, and the bubble wrap protection has been ruined, and it just doesn't work anymore, as the RA patients here on the call can testify. And so people have been trying to figure out what these cells are for a really long time. And so if you look in the medical literature, you see a lot of drawings that look a little bit like this. So this is supposed to be kind of like a knee, where the normal knee is on the left, and on the right, we see an inflamed knee, a cartoon of an inflamed knee that happens in rheumatoid arthritis. So in the normal knee, you're supposed to have this sort of bubble wrap, you know, kind of like loose, gentle, nice synovial tissue with a single row of cells called synovial cells or synoviocytes or fibroblasts. But then in rheumatoid arthritis, we have all these other cells coming into the joint. And so you can see how the cartoon shows the joint being swollen, being more red, more angry, and all of these different cells inside. And this is, you know, this is a great first approximation, but it's still clearly a cartoon. None of these cells are talking to each other. You know, they're just kind of like floating there in the ether. And, you know, this cartoon doesn't give us a great understanding about how these cells are working together to cause the inflammation that causes so much trouble in rheumatoid arthritis, because it's that communication between these cells that we know is sort of the best to target for new treatments. So, you know, over the last, especially 10 or 15 years, there's been new technological developments that have really allowed us to move beyond the cartoon on the left to new ways of looking at cells. And there's good news and bad news here. The good news is that it contains a ton of information. The bad news is it's really hard to convey to people who don't look at this all the time. So this graph here on the left, this is data from what's called a technology called single cell RNA sequencing. And so what's been done here is that tissue, synovial tissue, little biopsies or little tissue pieces from patients with rheumatoid arthritis have been collected with their permission to study rheumatoid arthritis and learn more about them. These tissue pieces have been cut up with like mechanical things and also with enzymes, so that each cell is now floating freely in solution. And then we put these cells on a special machine that looks at what each individual cell is making, so that we can tell what it is and what it's doing in that synovial tissue. And so what you're seeing here on the right is a graph where every single little circle, like this little circle and this little circle and this one, each one is a separate little cell. And based on what they're making, we can tell that some of them are fibroblasts, these sort of like bubble wrap kind of cells that are supposed to be there from the beginning. Some of them are B cells that make antibodies like rheumatoid factor or CCP antibodies. Those are here. Plasmablasts are a different kind of B cell. Monocytes or macrophages are these cells that come in and make lots of angry communication molecules and things like that. And then we have the T cells that I focus on down here. And this map, it doesn't show you where they are relative to each other in the joint. That's a separate issue that I'll get to at the end. But this sort of classifies them. It sort of puts them into categories so that we can dig deeper into exactly what they do. Because, for example, here among the T cells, you see that some of the circles are light green, some are dark green, some are brown, a few of them are yellow here if you look really carefully. Each different color symbolizes a different kind of T cell type. And the more data we get like this, the more fine-grained we can get. It's kind of like the baking aisle at your supermarket, where they used to have just like all-purpose flour, and now if you look, there's like tapioca flour, and there's gluten-free flour, and then there's whole wheat flour, and there's like so many different kinds, right? So there's the baking aisle, but then it gets more and more and more and more detailed. And the more detail we have, the more information we have, the more ideas we can get for new treatments. So let's look at the T cells, because that's what I study. So when we had this new technology, the single cell RNA sequencing, it showed us a new population of T cells that we hadn't seen before. And so this is a graph similar to the one that we just looked at. Each little dot, each green and pink and blue little dot is a different cell taken from synovial tissue from patients with rheumatoid arthritis. And we can classify these cells based on the things that they're making. That's what the single cell RNA sequencing technology allows us to do. The cells here that are in different shades of pink, those are CD4 T cells, which are a particular cell type that's been studied very extensively in rheumatoid arthritis, and for good reason. And so, I could talk for like an hour or two about the CD4 T cells and still not run out of things to say. But the surprise that we found is in this green and blue area here. These are CD8 T cells. So CD8 T cells are a type of T cell that we normally think about in viral infections. So if you get COVID-19, for example, your CD8 T cells are the assassin cells that come in and kill the cells that are infected by virus to help you defeat the infection. The same thing with influenza. CD8 T cells also kill cancer cells. So we think of these as like professional assassins that go around, find the cell that is wrong, either because it has a virus or because that cell is turning into a tumor cell, and then it kills that cell. And so, that's not really what's going on in rheumatoid arthritis. So people didn't really look at CD8 T cells all that much, but what it turns out that that's not what the CD8 T cells in rheumatoid arthritis are doing. So when we think about this sort of assassin kind of CD8 T cell, we do see a few of those, and we know what those assassin molecules look like that they make. And it turns out that these blue cells here at the bottom, this tiny little group of blue cells, those are assassin CD8 T cells. So they are present. But then you have this big group of green cells here that do not express the assassin proteins. These cells seem to be doing something else, and they're present in large numbers in rheumatoid arthritis. So the question is, what are those cells and what are they doing? So, all right. And so again, like the reason that CD8 T cells haven't been studied very much is because the typical function is for these CD8 T cells, they're even called cytotoxic T lymphocytes, cytotoxic meaning cell killing. So cytotoxic, assassin CD8 T cells, they release these killer molecules called granzyme B, perforin, granulysin, that go to the cell. So usually these are virally infected cells or cells that are becoming tumor cells, and they kill that cell through various mechanisms. And this has been, known for decades and decades. But this doesn't really happen in rheumatoid arthritis. And so that's why we hadn't studied it before. But again, those types of cells are only a small population in rheumatoid arthritis, but the bigger one expresses a little bit of killer molecules, but then a lot of this new molecule called granzyme K, or GZMK for short, because otherwise, I'm going to have to type too many letters. So granzyme K, what is granzyme K? So I've been studying immunology since probably 2001, and I had never heard this molecule before until I started working on this cell type about 7 years ago now. And a lot of people I meet who work in immunology also have never heard of this molecule, but I think it's actually quite important in rheumatoid arthritis and in a lot of other contexts. So let's look a little closer at this. So I'm going to save you a lot of basic science graphs and things like that to sort of tell you the summary of what I've found. And this is research that I did as an ANRF grant recipient a couple of years ago. And so this project would not have moved forward without the generous support that the ANRF provided. So I really want to thank this organization for that. And so what we've found is that granzyme K CD8 T cells, they are present in blood, but they're really abundant in rheumatoid arthritis synovial tissue. That's where they really like to go and they like to accumulate. And so they can travel into the bloodstream and then they can travel back out as well, we believe. These CD8 T cells can be activated in a couple of different ways. One is through the T cell receptor, which is the traditional way, but these T cells can also just eat cytokines. So cytokines are sort of the communication molecule that the immune system uses to signal inflammation. And these cells, they can be activated by that alone in order to start doing bad things in rheumatoid arthritis synovium. And that's pretty dangerous because it means that if they show up at any site of inflammation, they're going to start helping out to create more inflammation themselves. Through various experiments, we've discovered that granzyme K, this molecule that I talked about before, can act directly on cells in the synovial tissue. So this here is supposed to symbolize a synovial fibroblast, those cells that normally act like nice, kindly neighbors that form that sort of bubble wrap shell around the joint. If those synovial fibroblasts get the wrong kind of signal, get inflammatory signals, they turn into hungry, angry monsters, right? Just like all the other cells in the synovium. And granzyme K can do that through a couple of different mechanisms, leading to production of things like IL-6, CCL2, reactive oxygen species, molecules that we know are involved in rheumatoid arthritis inflammation. Some of you might recognize IL-6 because you might be on a medication like tocilizumab or Actemra, which is a medication that can target IL-6 pathways. And so we know that this molecule in particular is really important. These granzyme K CD8 T cells, they also release cytokines of their own. So again, cytokines are these communication molecules that act between immune cells. They make TNF. So TNF, again, that molecule that's inhibited by adalimumab or Humira, etanercept or Enbrel, and a whole bunch of other really useful rheumatoid arthritis medications. That cytokine is produced by these T cells, among other cells, and that also affects these synovial fibroblasts. And when you put TNF or interferon gamma or granzyme K together, then they act even more, so you get this sort of amplifying, snowballing effect from the granzyme K positive CD8 T cells on synovial fibroblasts. Now, this is one story, and this story was born out of a larger collaborative project involving medical schools across the country, rheumatologists and scientists across the country, and even in the UK, called AMP, the Accelerating Medicines Partnership, which assembled this team of, probably about 100 different rheumatologists and scientists who collected synovial tissues and then lupus nephritis kidney biopsies in order, again, to learn more about the cells that are present in the joint. And so the granzyme K project that I told you about, in the last few minutes was born out of the first phase of this. but I've also been directly involved with the second phase, where again, we're digging deeper. We're looking more in more detail at that baking aisle to figure out exactly, all the different kinds of flour that we can find, bread flour and enriched flour and unbleached flour and like every single thing, so that we can have the most detailed understanding of what's happening in rheumatoid arthritis joints, so that we can dig deep and dig hard to find new treatment targets to help patients who are struggling with rheumatoid arthritis. And so, you might remember that map that I showed earlier that had some like blue and it had some green and brown and things, and there were like a few dots. Now with new technology, so again, technological developments are helping us. Here we have 95,000 T cells from rheumatoid arthritis patients. This is from about 80 different patients with synovial tissue combined together to find all these populations. So before, you remember there were 3 shades of pink, and then there were green and blue cells. So we only had 5 different T cell populations. Now we have 23. And so we're learning more and more and more about the T cells in the rheumatoid arthritis synovium. In the same project, we're learning about the macrophages, about the fibroblasts. We're now learning about the blood vessels, which we haven't seen before, because it turns out the blood vessels really need to grow to create all that swelling and redness and heat that you feel in joints that are swelling and that are flaring. And so the endothelial cells also play a role in flares. We have NK cells, we have B cells. We're sort of swimming in information that this big consortium, this network, a team of different institutions has generated. And now we get to spend the next few years, digging into this data and finding new things and looking for new mechanisms and connections. And so that's been really inspiring to be a part of. And one thing we did with this information, I mentioned that this is from about 80 different rheumatoid arthritis patients, is that we realized that rheumatoid arthritis is not just one disease. Like if you have friends who have rheumatoid arthritis, you might have realized that your rheumatoid arthritis and their rheumatoid arthritis might affect different patterns of joints. Maybe they don't have foot involvement at all and only have the hands, whereas you also have like knees and elbows and all those kinds of things. Or, like some people respond really well to TNF blockers, right? And other people do not. They need tocilizumab, for example, like the IL-6 blocking medication that I mentioned. And so we already knew that there were categories of rheumatoid arthritis. Now, when we look at cells, we see these categories. So here, each column is one patient. And the column here is shaded by the different kinds of cells. So blue is blood cells or endothelial cells. Light blue is fibroblasts. The sort of salmon colored pink is myeloid cells or macrophages. Purple is NK cells and light purple is T cells. And so you can see that some people have sort of a mix of all these things. Some people are really, really salmon pink. They have a lot of myeloid cells and not a lot of other stuff. And then some people have, lots and lots of these like lavender T cells with some myeloid cells, like all these different kinds of combinations of cells. And it turns out that determines the kind of molecular environment that's present in their rheumatoid arthritis, which could also, we're hopeful that this kind of categorization will help us better predict who's going to respond to what treatment, right? Because I'm sure that many of you had to try several different things before something worked. Like, what if we could do a blood test or a synovial fluid test or even a synovial fluid biopsy in order to help you jump directly to the treatment that's going to work instead of spending months or even years trying things that never help you. And so, so we're starting out with this sort of cartoon image with just a few cells like floating around. We're starting to put these cell types together and understand how they're using cytokines, these sort of communication molecules, to talk to each other. And that helps us also to identify how we can block this communication to stop them from talking to each other, because that's how you stop inflammation from perpetuating. And so we have, this is the far left image is from 2016. So only 7 years ago, this was the best we could do. Here's a review that came out in the New England Journal of Medicine earlier this year. And, you see a lot more detail, a lot more dynamics about how these cells are interacting. And, I think that pretty soon we're going to have a hard time publishing our diagrams of how these cells interact on one single paper, because there's going to be so many different cell types and pathways. But that's the goal, is to outgrow a single piece of paper in our understanding of how these cells interact together. And so I want to thank my mentor in Boston, who I worked with for several years before moving out here to Colorado, Michael Brenner, and several members of the lab listed here. I am not a computer scientist, a computational biologist. And so a lot of this work with those like little cells and the different colors was done in collaboration with Fan Zhang, who is also an ANRF recipient. And she was in Boston, and now we're both out here in Colorado. Fan Zhang's mentor in Boston, Shomer Chowdhury, has also been a great help with this and has also played a central role in the Accelerating Medicines partnership. And I especially, again, want to thank the ANRF for funding, because without it, this project would not have moved forward at a very crucial state. And with that, I'm happy to take questions.
Thank you very much for that, Anna Helena. That was a lot of really good, informative information that really makes sense for those of us that have dealt with RA for quite some time. Again, put some clarity to the signs and symptoms and why we have things going on that are happening to us over and over again. With your discoveries, what does this mean, or what do you see this meaning for, early treatment and possibly even a cure of rheumatoid arthritis?
Yes, I think, I think we're edging closer to a cure, but I think we're still like a few miles away, unfortunately. But every discovery we make puts us closer. I think the other thing that's really neat here in Colorado and at other institutions is that they're looking at pre-RA. So Nancy, you mentioned that sort of, that the sort of like vague symptoms that happened early on, and you might have even like, in hindsight, recognized even earlier symptoms that were probably the premonition of all this happening. Can we identify people there and treat them with something simple to stop the full RA diagnosis from happening? So, I think prevention is going to be easier. Preventing RA from developing will be easier than curing it once it's here. But, both are goals of the rheumatology research community. In terms of like what I think that, so I think that the project that I talked about at the end, this Accelerating Medicines partnership, deep dive into 80 samples and classifying people, I think that could be really key to people with new diagnoses, because I think that is the time when you spend, often months with your rheumatologist trying to find a treatment. And each one, it takes 2 or 3 months to figure out if it works. And so you try one and then you try another. And even if you're going at the fastest pace you can, it takes, often a year. And so if we could skip that part and go straight to the treatment that works for you, one, it'll prevent that 1 year of terrible suffering before you find an effective treatment. Two, just as Dr. Chang mentioned, the sooner you get rheumatoid arthritis under control, the better your lifetime outcome will be in terms of joint function, in terms of heart disease, in terms of everything. And so, we're all really focused on getting people the effective treatment they need as quickly as possible. And we need to figure out how to know which treatment will work. And that's been something that the research community has been struggling with. But I think these new cellular classifications could be the key.
Great information. And just from my own perspective, that I wish I would have known some of these things when I was first diagnosed, to hit it hard, hit it fast, and stop progression.
I think it's often tempting, and I have a lot of patients who say like, well, like I think I want to try some like natural remedies or things like that. And I believe that patients have autonomy. Like you get to decide what happens to your body, but there is sometimes there's a big cost down the road that you don't realize you're going to have to pay until after the fact. And I wish we could do better at preventing that.
Yeah, I agree. I've been victim of that my own. What role do you think hormones play in RA? And it seems one of the questions is, it seems like more women than men get autoimmune diseases. Some women flare after pregnancy. Some women are diagnosed when they come off the pill or in menopause. It'd be interesting to know how hormones fit into this whole picture of rheumatoid arthritis.
I think that that's something we're starting to sort of learn more and more about. So, just like the person who asked the question said, rheumatoid arthritis is much more common in women, about like 5 to 10 women per man among the whole population with people with rheumatoid arthritis. And that's not true for all cases of autoimmune disease. So for psoriatic arthritis, for example, which looks pretty similar, like some of you might have had a rheumatologist who wasn't really sure if you had psoriatic arthritis or rheumatoid arthritis at first. Psoriatic arthritis is about equal between the genders, might even be a little bit more like leaning towards like more men than women. And so like what causes that? I don't think we have a great understanding. We do know that men's and women's bodies function a little bit differently when it comes to the immune system for a few different reasons. I think one of the biggest one is that women are pregnant. And so the child that you carry, the fetus, is genetically different from you. And so to prevent you from rejecting that as something that shouldn't be there, like a cancer cell or a virally infected cell, the female body has to make certain adjustments, especially during pregnancy, that then sort of revert afterwards. And so, just like you say, a lot of people are diagnosed after they give birth, probably as part of those like hormone shifts that switch from tolerance during pregnancy to sort of the normal state again afterwards. And so I think this is a work in progress. There are a few other leads and possibilities that are probably all sort of working together to generate that female predominance in rheumatoid arthritis.
Thank you for that. Another question we have from someone watching is, what have studies shown? Is there a connection between leaky gut and the development of RA or triggering of flares?
Yes. So what like between the gut and flares. So like the interesting thing about rheumatoid arthritis is the more that we learn, the more we think that rheumatoid arthritis doesn't actually develop in the joints. It probably develops either in the mouth or in the lungs or in the gut, where sort of faulty interactions between the immune system and the bacteria that are naturally present in those sites kind of goes wrong, and then creates this immune response that then cross reacts with the joints, and then you start reacting to the joint tissue instead. And so I think in terms of the gut, there are fewer connections between the gut and rheumatoid arthritis than there are with psoriatic arthritis, for example, which often happens together with ulcerative colitis or Crohn's disease, these sort of official autoinflammatory and autoimmune diseases that happen in the gut. But a lot of people with rheumatoid arthritis describe feeling better by, trying to reduce the amount of gluten in their diet or trying to reduce the amount of red meat in the diet. And that's probably because of just sort of regular irritation that some people have more than others. Red meat, for example, is known to be sort of just generally irritating towards the gut. And so, switching to a vegetarian diet can help. I think I do want to urge caution if you're planning on changing your diet, make sure to discuss anything that you do with your primary care doctor and your rheumatologist, because you do want a well-balanced diet, right? So spent many years in Boston. Some of you might know Tom Brady and his like completely insane diets. Don't do what he did. Like you want to eat tomatoes and other vegetables and all these kinds of things. But do feel free to ask your rheumatologist about diet if you feel like GI symptoms are correlating with your arthritis symptoms.
Perfect. How can people stay informed about the latest research developments in our disease?
Yeah, well, I think you're doing a great thing by attending this seminar, right? So I think that this is sort of like hot off the presses kind of information from the cutting edge of science that the Arthritis National Research Foundation is providing right here. The other thing you can do is follow the American College of Rheumatology. It might sound like a university, but that is actually the National Society for Rheumatologists. And so just here in like about 2 or 3 weeks, about 10,000 rheumatologists will be gathering in San Diego to share their findings, to discuss the latest developments in rheumatoid arthritis treatment and research and so forth. And the American College of Rheumatology does its best to sort of convey this to the patient population as well. You can also look into getting involved with local organizations, arthritis foundations and things like that, and sort of keeping track like that. But I think that the ANRF has been really like leading the way in terms of sharing research findings with the patient community.
All right, so as we wrap up, I wanted to say thank you so much to our fantastic moderator, Nancy. We so appreciate you being here and sharing your story. Also to our wonderful researchers for sharing their innovative work towards understanding rheumatoid arthritis. You can really see firsthand the impact ANRF has had in supporting these incredible individuals in their quest for a cure. I'd also like to thank once again our partners, Amgen, AbbVie, AstraZeneca, and AOPI for your support of this webinar. And lastly, we'll be sending our live attendees new disease fact sheets along with the survey after the event. Your feedback really helps us find direction for future webinars. Please share your comments with us so we can continue providing important and relevant content to you. And please don't forget to sign up for our other Researcher Spotlight series. We will be live at the same time on November 1st with the juvenile arthritis webinar. So thank you so much to everyone who joined us for this great event and for all your wonderful questions. If you enjoyed this webinar, we kindly ask that you consider making a donation today by visiting curearthritis.org/donate. Every contribution helps support grant funding for brilliant researchers like the ones you saw here today. Thank you all so much for coming, and please remember to follow us on social media to stay connected.